Hi biologists. Let's start by looking at the learning objectives for this lesson. At the end of this section, and following the biology syllabus, take note that this material is for the higher level biology only. You should be able to use the Punnett square technique to study the inheritance to the second filial generation, F2, of two unlinked traits. What does this actually mean? What are we trying to learn? Well, we have to be able to use the Punnett square technique to study the inheritance of two unlinked characteristics or traits to the second generation or to the second filial F2 generation. We now need to be able to explain Mendels Second Law based on chromosomes. Don't forget that a gene controls a characteristic, and each gene has two different forms or two different alleles. So do you recall that we have a gene for height in pea plants, and the gene for height has two different forms. One allele is big T, which is for tallness, and the other allele is small t, which is the gene or the allele for smallness. One allele is dominant and it masks the effect, or it bullies the effect of the other allele, which is recessive. Two alleles, or two forms of the gene, are needed to control the outward appearance or the expression of the gene. So to be tall or to be small, you have to have the two alleles. Homozygous means that the two alleles are the same. So if you are tall, your genotype or the type of genes you would have are represented by big T. And if you are small, your genotype or your genes would be represented by small t and small t. Heterozygous means that the two alleles are different from one another. So if you're a pea plant and you are tall and you are heterozygous, then you are big T, small t. Don't forget, of course, the word homo means the same, whereas hetero means different. Now, I find it easier to understand chromosome behavior in gamete formation if you imagine you have 4 sweets in your hand, 2 blue ones and 2 green ones. They happen to have letters written on them, rather like M&Ms. Now, when you write letters in genetic crosses, you must make sure that you distinguish the letters one from another, that you distinguish the capital letters from the small letters. That is why I have the letter Y underlined here to show that it is a small letter Y and to distinguish it from capital letter Y. Now back to the sweets. Imagine you've been told to half them with your brother or sister. What are your options? Well, you might start with 4 sweets, but you're going to half them, and your sibling is going to end up with only 2. So what are your options? Well, they're rather fussy, so they only want 1 blue sweet and 1 green one. So what combinations can you give them the sweets in? Well, you could give them the blue sweet with the capital letter T with one of the green ones with the capital letter Y. Your other option would be to give the blue sweet with the capital letter T and the green sweet with the small y. Another option would be to give them the blue sweet with the small letter T and the green sweet with the big letter Y. And your last option would be to give them a blue sweet with the small letter T and a green sweet with the small letter Y. There are your 4 options. Of course, this is representing chromosomes. So therefore, this is a heterozygous parent. A genotype, or the type of genes that it has, is big T, small t, big Y, small y. We are going to form gametes. We're going to half the number of chromosomes. So when you're halving the number of chromosomes, we end up with 4 options. We end up with big T, big Y, big T, small t, big Y, small t, small y. Now, this is the understanding of chromosome behavior in gamete formation. But we do have a trick that we can make use of when we're doing genetic crosses. I call this the arrow trick. Basically, if I take my big letter T and draw an arrow out into big Y, I can create a gamete that contains big T, big Y. I can draw another arrow out of the big T and go into the second letter of the Y's, and then I will create the gamete big T, small y. I can draw an arrow out of the small t into the first of the y's, and then I will create the gamete small t big Y, and I can draw an arrow out of the small t into the small y and create the gamete small t small y. So using the arrow trick, you can see that I end up with the same 4 options. And we make use of that trick when we're doing our genetic sums, as I call them, or your genetic crosses. Now let's represent this by means of a chromosome diagram. Here we have a pair of homologous chromosomes in blue in charge of the characteristic of height in the pea plant. We have 2 alleles for the gene for height. We have big T representing tallness and small t, the allele for smallness. We have a pair of homologous chromosomes here in green for the characteristic of pea colour. The gene for pea colour is represented by 2 different forms, big Y for yellow and the allele small y for green. Now this parent is heterozygous, so we have the possibility of getting 4 gametes. Remember our arrow trick? The first gamete will contain a big T with a big Y. The next gamete will contain a big T with a small y. The next gamete will contain a small t and a big Y, and the last gamete, the last possibility, is that it will contain a small t and a small y. Stating the facts, the chromosomal basis of Mendels second law is that when gametes are formed, either of a pair of homologous chromosomes. So our pair of homologous chromosomes was big T, small t. So either of them, the big T or the small t, can wind up with either chromosome of the second homologous pair. So in other words, the big T or the small t can wind up with the big Y or the small y in the second pair. Now let's look at a dihybrid cross between 2 homozygous parents. Now the parents' physical appearance are that they are homozygous, both of them. You have a pure breeding tall yellow pea plant crossing with a pure breeding small green pea plant. Here are the parents' genotypes. What type of genes do they have? Well, because they're homozygous tall and yellow, it can only be big T, big Y, big Y. The other plant is homozygous again. It is small and green, so it can only be small t, small y, small y. Now, the gametes that we can get from each of these parents is that we have the possibility on meiosis of getting 4 gametes. Using our arrow trick, the gametes would be a big T with a big Y, a big T with a big Y, a big T with a big Y, and a big T with a big Y. So strictly speaking, the 4 gametes here all happen to be identical to each other. From the other parent, we get the same thing. We have 4 possible gametes, a small t with a small y, and a small t with a small y, a small t with a small y, and a small t with a small y. So basically, in a nutshell, all the gametes are the same as each other, all small t, small y's. So it doesn't matter what gamete will fertilize the gametes from the other parent, you will always end up on fertilization with the same result in the F1 generation. The genotypes that you will create will be that they are all big T, big Y, small y. What is their physical appearance? Well, all that first generation will be tall and yellow. Now, what will happen if we let this F1 generation self-fertilize? Before we go anywhere, this should be ringing a bell. Let's show how Mendel was vindicated. These results show that Mendel was perfectly correct when he got all the F1 generation to be tall and yellow-seeded pea plants. When we self the F1 generation, we actually end up with a dihybrid cross between two heterozygous parents. The parental phenotypes are that both parents are tall and yellow. The parental genotypes are that both parents are heterozygous, big T, big Y, small y. Now, upon meiosis, we should know that we have the possibility of getting 4 different gametes. Using our arrow trick, we can demonstrate this. One gamete will be big T, big Y, the next gamete will contain big T, small y, the next gamete will contain small t, big Y, and the next one will contain small t, small y. We get the same from the other parent. So we have 4 possible gametes from the parent on the left and 4 possible gametes from the parent on the right. How will these combine on fertilization? Well, to show their possible combinations, we make use of what we call our Punnett square, which we have met before. Now, as we have said, it doesn't matter what gametes we put along the top or along the side, it doesn't make any difference. So the possible combinations for the F2 generation as a result of these fertilizations is that big T, big Y gamete will fertilize a big T, big Y to create a possible offspring of big T, big Y, big Y. You might notice the convention or the habit that the T's are written together first and the Y's are written together second. If this gamete is fertilized by the other gamete here, big T, big Y, then this will lead to the possibility of an offspring, big T, big Y, small y. Working our way through the Punnett square, we will realize that we will get 9 tall yellow pea plants, 3 tall green-seeded pea plants, 3 small yellow-seeded pea plants, and 1 small green-seeded pea plants. Where did we hear that before? Again, let's show how Mendel was vindicated or proved to be right. These results show that Mendel was perfectly correct when he got 4 different types of F2 plants: tall and yellow, tall and green, small and yellow, small and green, in a ratio of 9:3:3:1. He had done it all before. What are the expected ratios from Mendel's second law that we should make it our business to know? Well, if there's a dihybrid cross between 2 homozygous parents, then all the offspring will be the same as the dominant parent. In Mendel's example, when we crossed a tall yellow-seeded pea plant with a small green-seeded pea plant, the F1 generation were all tall and yellow-seeded, the same as the dominant parent. If there is a dihybrid cross between 2 heterozygous parents, in Mendel's case between the tall yellow-seeded pea plants of the F1 generation, the offspring will always be in a ratio of 9:3:3:1. Now, why is this the case? Well, we have to have these facts at our fingertips. We get these ratios because there were 4 possible gametes from the heterozygous parent and the genes for both characteristics were not linked. In other words, the genes for height, the alleles big T and small t, were on different chromosomes to the genes for pod color, big Y, and the allele small y. If the genes were linked, we would get different ratios. The law of independent assortment also took place, meaning that the pair of alleles or genes for height separate from each other during gamete formation independently of the pair of genes or alleles for pea color. The T's separated, sorted themselves out independently of the Y's who separated and sorted themselves out. So in other words, any one of the genes for height could end up with either of the alleles or the genes for pea color. Now that we have reached the end of our lesson, have we achieved our objective? Can you use the Punnett square technique to study the inheritance to the second filial generation, F2, of two unlinked traits?